Welcome to The Explainer. Today, we're going on an incredible journey from a macro-level mystery that you've probably wondered about, zooming all the way down to the microscopic, atomic machinery operating inside your cells at this very second. We are unlocking the biological operating system of life to understand the sheer mathematical impossibility of you. Okay, let's dive into this.
You know, it's a deeply personal question, right? Assuming you aren't an identical twin, you and your siblings come from the exact same parents. You were built from the exact same two genetic lineages. And yet, you have different eye shapes, different heights, different immune system strengths, and even different health risks. How is it that the exact same genetic starting materials produce such wildly different human beings?
While the answer lies in the numbers, right now the chances of two siblings inheriting the exact same genome from their parents are less than 1 in 70 trillion. Just let that sink in for a second. 70 trillion. That staggeringly huge number is the direct result of an incredible microscopic reshuffling power happening inside the human body. It's a specialized process designed for one thing: to ensure that no two humans are ever constructed exactly alike. And this brilliantly illustrates the difference between how our bodies grow versus how they reproduce. First, we have mitosis. Think of this as your body's cloning engine. It's how a skin cell copies itself to heal a paper cut, generating two genetically identical daughter cells. But then we have meiosis. This is a highly specialized reductional division strictly designed for sexual reproduction. It takes a standard cell and cuts its chromosome count in half to produce gametes, sperm or egg cells. But more importantly, as it does this, it completely scrambles the genetic code to guarantee that every single gamete is entirely unique. The first major mechanism driving this uniqueness is something called crossing over. Discovered by Thomas Hunt Morgan back in 1911, a massive finding that actually won him the Nobel Prize. Crossing over is basically the ultimate biological shuffle. It is the literal, physical breaking and swapping of DNA segments between maternal and paternal chromosomes. This incredible process breaks old linkage groups and creates brand new, never-before-seen combinations of alleles on a single chromosome. Let's actually look at how this swap happens during a phase called prophase I. Think of it less like an abstract biological concept and more like a fascinating mechanical factory. First, your maternal and paternal chromosomes find each other and pair up side by side in a process called synapsis. Next, an incredible structure called the synaptonemal complex acts exactly like a microscopic protein zipper, locking them tightly together into a 4-part structure called a tetrad. Then, the non-sister chromatids break at perfectly matched locations called chiasmata. And finally, they physically trade pieces of code. The maternal chromosome ends up with paternal tips, and the paternal chromosome gets maternal tips. They literally rewrite each other.
But hey, the uniqueness engine doesn't stop there. Enter mechanism number 2, independent assortment. When those newly shuffled chromosome pairs line up in the middle of the cell to divide, they do so entirely randomly. The orientation of one pair has absolutely zero effect on the orientation of another. In a human cell with 23 pairs of chromosomes, this random lining up alone generates over 8.3 million possible gamete combinations from a single person. So when you combine 8.3 million combinations from one parent with 8.3 million combinations from the other, and then add in the wild card of crossing over, well, that is exactly how we reach that mind-blowing 1 in 70 trillion chance of identical siblings.
Section 1. Decoding the double helix. Let's move to and see how this builds as we zoom in from those massive dividing chromosomes right down to the actual molecular code itself. This brings us to the architecture of the DNA double helix, and specifically Chargaff's rules. Our genetic alphabet basically has 4 letters: adenine, thymine, guanine, and cytosine. But here's the catch: they aren't all the same size. Purines, which are adenine and guanine, have a bulky 2-ring chemical structure. Pyrimidines, thymine, and cytosine only have a single ring. If 2 bulky purines paired together, the DNA double helix would bulge out. If 2 pyrimidines paired, the helix would just collapse inward. By strictly pairing a 2-ring molecule with a 1-ring molecule, A to T and G to C, the DNA maintains a flawless, uniform 2.0 nanometer width down the entire length of the helix. It's an absolute architectural masterpiece. And the length of that flawless helix is just staggering. Inside the microscopic nucleus of every single cell in your body, you have about 2 meters of this molecular code. If you took all the DNA inside your body right now and stretched it end to end, it would reach the sun and back 60 times. That's enough to literally boggle the mind. And this massive, intricate code is being perfectly managed, regulated, and read inside you at this exact second. Section 2. Turning genes on and off. Because having a unique DNA blueprint is really only half the story. Uniqueness also stems from how your cells regulate that exact identical code. So the crucial point here is epigenetics. Eukaryotic cells use chemical markers that act kind of like molecular light switches to tightly control which parts of your unique blueprint are actually read. For example, histone acetylation adds acetyl groups to the proteins your DNA wraps around. This relaxes the structure, efficiently turning gene transcription on, making that code accessible. On the flip side, DNA methylation attaches methyl caps directly to the DNA bases themselves, aggressively silencing gene transcription, locking it off. It's a highly sophisticated control board that ultimately dictates your cellular identity. Section 3. Editing our unique code. To truly understand this biological frontier, we need to zoom back out and look at a monumental shift, the era where humanity has actually learned to intervene. Just look at this incredibly rapid timeline of biological mastery. In 1953, Watson and Crick published the structure of the DNA double helix. At that point, we were merely discovering the shape of the code. Fast forward to 2006, and Shinya Yamanaka discovers that by injecting just 4 specific factors into a fully mature adult skin cell, he could completely wipe its epigenetic memory, reverting it back into a virgin stem cell. And then, in 2012, Doudna and Charpentier developed CRISPR-Cas9, utilizing a bacterial defense system as molecular scissors to cut and edit DNA with atomic precision. Today, these tools are already in clinical trials treating genetic blood diseases like sickle cell anemia by physically correcting the mutant code in living patients.
Now, what's really interesting about this reality is the profound philosophical shift it brings. When we look at mitosis, meiosis, DNA replication, and gene editing, we aren't just looking at abstract science. You are not merely observing life. You are a conscious, self-aware collection of 37 trillion cells that has somehow learned to decode, read, and manipulate its own genetic source code. You are literally the biological universe experiencing itself.
Which leaves us with the ultimate question as we look ahead. We've conquered the shuffle, we understand the mechanics behind our 1 in 70 trillion uniqueness, but what does it mean for our future as a species now that we hold the molecular scissors, now that we can read, reprogram, and rewrite the very code of life itself? Keep exploring this molecular frontier, and as always, stay curious.